Get started, I just want to thank really the CTSI and all of their support for this amazing lecture series. And my name is Dan Geschwind, and I'm the director of the Institute for Precision Health, as well as in the Department of Neurology and Psychiatry and Human Genetics here. And it's really my great pleasure to have the opportunity to introduce Eran Halperin, who's a professor here in the departments of computer science, computational medicine, anesthesiology, and human genetics. He's also the senior vice president of AI and machine learning at Optum Labs, which is part of United Health Group. I'll tell you a little bit about his training. He's always been a math and computer scientist. So prior to what he's doing now, he got his Ph.D. in computer science from Tel Aviv University, working on optimization problems, and that was in 2001, following both an undergrad and master's summa cum laude in math and comp science. He's held a lot of research positions and postdoctoral positions at places in California and elsewhere, including UC Berkeley, the International Computer Science Institute at Berkeley, Princeton University, and Tel Aviv University, where he was chair of bioinformatics before we were able to recruit him here. His lab has developed methods solving a lot of different problems that have appeared in genetics and genomics, but he's really focused the last half dozen years in his lab on health-related domains. So he's worked on the population genetics, methylation, microbiome, single-cell RNA sequencing, developing software for deconvolution of bulk tissue, et cetera. The work that he started to do here and that he's been doing now elsewhere as well is in medical applications. So working in medical imaging, looking at waveforms and combining that with EHR and genomic data. So, and in fact, the work that I think he's going to talk about today involves work that he did here at UCLA with methylation data, in connecting it to our EHR. Eran has a lot of awards. He's published more than 150 peer-reviewed papers, and really has gotten some of the highest honors to scientists in Israel, including the Rothschild Fellowship, the Technion Jaludin Prize for Technological Contributions to Medicine, the Krill Prize, and he's elected as an International Society of Computational Biology Fellow. So it's great to welcome Eran here to share his work with us today. Thanks, Eran.
Thank you, Dan, and thank you for inviting me. And so I will share my screen, I guess. Okay. So it's great to be here in person. I'm really an advocate for coming back to the in-person culture, and kind of miss it. So I basically wanted to give this presentation in person, and hopefully we'll also get some questions and interaction with the people on this over the Zoom. So as Dan said, I basically kind of looked at different over my career. I looked at different applications in medicine that include things that are related to genomics, but also recently things that are related to applications in medicine, including medical imaging and physiological waveforms in the EHR. I'm going to talk today about a kind of a combination of the EHR and genomics. And as Dan mentioned, that's work that we've done here in the lab. I want to start with just kind of giving an overview on about like the things, kind of the vision of basically the things that we're looking at. This is true both in the lab and also in my role in United Health Group. So let's see, why doesn't it move? There you go. So the vision or the kind of the motivation for a lot of our work is basically trying to find signals in the data that allow us to do better diagnosis and have better prognosis of disease, or kind of predict the prognosis of the disease And then ideally that should lead to better treatment. So this is, not something that is specific to what we're doing, but really everything that we think about is basically starting from that kind of point of view. There's many different things that can go into these models, including medical history, family history, the symptoms, clinical labs, and basically a lot of types of data that would go into the medical history, including what we mentioned before. So waveforms and medical imaging and genomics. And so there's some questions here about what is useful in what to what applications. So some applications, the genomics is going to be the best thing to look at, and so for other applications, the clinical labs, and other ones, it's going to be the imaging. Eventually, if we had some kind of a machine that basically would know exactly which every molecule in our body, where is it, and how much of this do we have, then basically all of this is going to converge to one thing. But right now we're looking at essentially proxies to this kind of picture, and we're trying to see how are these things could be related to prognosis and diagnosis of disease. So before I start going into the subject of this talk, I just wanted to give kind of more of an overview of other things that we're doing, and I'm going to give a couple of examples here. And one of them is a recent paper that we had on prediction of continuous blood pressure based on ECG and PPG. So what you see here on the right is basically the top one is an ECG waveform, and the middle one is a PPG waveform. And you're trying to take these two waveforms and predict what's going to happen to the continuous blood pressure. And the continuous blood pressure is something that is being measured in the ICU or in the OR, just to make sure that basically there's no drop in blood pressure or something like that during the operation. So this is again an example of a different type of data that could be useful for, in this case, that would be diagnosis of something in very short time frame. Another example is a recent work that we did on AMD and on the staging of AMD, so age-related macular degeneration. This is actually a disease that is one of the first GWAS basically was done on this, because there's a lot of genetic components to the disease. But over the last 10-15 years, there's been a lot of usage of OCT. So this is three-dimensional imaging of the retina. And the question is basically, can we use this in order to figure out whether someone is deteriorating, or basically what's the progression of the disease? So it's not really just a kind of a 0 and 1. It's not that you don't have AMD and you do. Actually, if you take all of us when we're 90 years old, probably we all have to some degree AMD. The only question is how bad it is and how severe it is, and the slope, essentially, how fast do we progress? So these two examples are, this one, the previous ones are using essentially deep neural networks. In this case, it's a deep neural network that we designed that is specifically for three-dimensional images. So, and then as I mentioned, basically, I'm also working with a United Health Group, and over there we're looking more at the things that are the imaging and the EHR and the wearables, and less actually at the genomics, and basically trying to use all this to improve the prediction of diagnosis and prognosis of the patients. Okay, so in this talk, I'm going to talk about EHR, electronic health records, and as you probably all know, in many different places right now, the EHR is in many medical institutions. EHR is now very ubiquitous. Basically, essentially, all the big places would have them, at least in the US. And here we collect in UCLA, as an example, we have the EHR for the last 8-9 years through the EPIC system. And one thing that anyone that is basically looking at the EHR and tries to use this data in order to predict what's going to happen to a patient, or what happens to a patient right now, one thing that you very fast we'll see once you look at the data is that it's very sparse. There's a lot of sparsity in the data, and there's a lot of missing data. And that's very different from genomics. I know that we're looking at imputation in genomics, but still in genomics, we usually basically look at the entire, when we sequence the entire genome, when we look at the expression level of across the genes. So we may have some inaccuracies in some places and some ambiguities, but generally we have the information across the entire genome. Here, in many cases, we're just not going to have this information for some of the patients. Now, another complication in EHR is that the data is not really missing completely at random, which is typically what you'll have in other applications like the genomics that I mentioned before. So for just as an example, basically, if you think about a clinical lab that is missing for a specific patient, it is often the case that is missing just because that patient doesn't have family history or medical history that corresponds to that clinical lab. So the information about that patient, the other information about the patient, is related somehow to the missingness. So it's not that basically something just went wrong and the clinical lab would just dropped on the floor or something like this. There's something that is more inherently structured in the data or in kind of the generation of the data that gives you this missingness. And that basically means that if you just try to use the kind of typical imputation methods, the typical imputation methods that use maximum likelihood EM, they would assume that everything is completely random. And this assumption is not correct here. You can still do this, but you're going to induce some biases that you kind of want to avoid. So one way to deal with it is to add additional information and making it basic. Once you add this information, then the data becomes missing it completely at random. So one type of data that we could potentially use is genetic data or genomic in general, but let's discuss genetic for the next few minutes, and then we'll go to other types of genomic data. So you could potentially look at genetic data. So let's say that we had whole genome for each patient, and then you could try to use any method that you want to predict what is each of the values in this matrix here of the patient. So that has been basically, many people have been looking at this, including here at UCLA, but really across the entire community, and trying to develop these methods that we call them polygenic risk scores, which is essentially a linear model that basically tells us what is the risk for each patient based on kind of what are the positions where that patient has a variant. So you just take a linear model across many different SNPs or variants, and then you accumulate them. You give each one of them a value, an effect size. In this case, these are these AIs, and then you get a number per person. You basically hope that if a person has a higher number, that's indicative of something. So, for example, that patient has that condition. So you can look at the distribution of the PRS across the population. And again, the hope is that if you look at the high risk population, so if you look at the patients that have the large numbers of the PRS, these patients are going to be, there's going to be a kind of an enrichment of patients that have a certain disease for which you do the PRS. Now, one of the things that you could do is basically then try to connect the genetics to the EHR, and this is one way to do this is using the biobanks. So the biobank that we have here is the Atlas biobank that basically the Institute of Precision Health is generating. And so I don't know the numbers that we have right now, but the idea is to have 150,000 at some point soon. And patients where we have a whole genome genotyping for them, and we can then connect these genotypes to the medical records, so to the EHR, and potentially do what I just showed in the previous slide. Now, obviously, this is not the only biobank. There's the UK biobank that many people are using that And there's other institutes that are also generating their own biobanks. But we actually used, and I'm going to talk in this talk about some of the, what we did here is we took the UCLA biobank, the Atlas biobank, and we took some of the patients from there and we added some additional measurements, and we tried to see if this can be useful for the imputation. Okay, so genetics and PRS is a great start, but it does have some limitations. And I listed here some three. It doesn't have, not supposed to be a comprehensive list. Maybe there's others, but these are kind of three major ones that I think that we have to think about when we use PRS or when we use genetics. So one thing is that the genetics doesn't capture environmental factors. So essentially, basically, you're born with your genetics, and it doesn't change by the environment. It doesn't. There is no correspondence to the environment. And when I'm saying environment, it could be the pollution or something like that, but it could be also your diet, or it could be anything that, or medication that you take, anything that basically affects you from the outside. Genetics is static. This is related, but basically it doesn't change over time, which essentially means that whatever we can predict using genetics, we can predict it when you're born, right? So it's not that when you're 50, we can predict a different thing than when you're 30. So in some cases, it may be good enough, so especially if the heritability is very high. So think about a disease that the heritability is very high, in that, or maybe 100%. So it's completely determined by the genetics. Then in this case, genetics is all you need. You don't need the environment, and it's not going to change over time. Think about another condition that is 50% heritable. Then you'll be able to say that that patient has predisposition, but you're not going to be able to measure the effect of the environment over time. And so you'll need other type of information, which people typically do. They use age and they use other type of covariates on top of just the genetics to kind of account for this. But just the genetics by itself doesn't give you this information, the full information. It's more than that. Sometimes you'll know that maybe the disease is relatively highly heritable, but the age of onset is not necessarily something that you can predict using the genetics. So if I want to know just the 5 years risk for a specific patient, then I need not only that the condition would be highly heritable, but also that the age of onset would be. Again, this is only if you're looking at the genetics in isolation. If you start adding other types of information, you could potentially improve this. And then the third point is that, and this is, not necessarily science, it could be science plus other things that come into kind of the decision of what to introduce into medicine. It just basically, if you look at all the effort, there's been quite a bit of effort in the community. It's not that you go today to your primary care physician and they ask for, a test that will do a PRS for you. It's very far from this. Actually, it's not very widely used. And so that basically means that there's more to explore. It doesn't mean that we shouldn't, that we should stop. There's more to explore on the third point here. And we, as a kind of community, we should try to basically push that more of this actually will go to the medical practice. So what I want to talk today about is basically how do we complement this information, or how do we kind of compare that information that we get from the genetics with information that comes from methylation. So let me give a brief introduction to methylation for those who are less familiar. So methylation is basically, actually, I'm going to go back one slide before I do this. So methylation is essentially methyl groups that are basically connected to specific positions in the genome. And so unlike genetics, where essentially every cell has the same DNA, the same genetics, in methylation, in every cell will have different positions in the genome that are going to be methylated. And so that would be different between different individuals, but also between different cells. There's some information that we know that's been, for the last maybe 10 years, it's been studied quite a lot with respect to diseases, with respect to other types of traits and conditions. And there's some things that basically show that that methylation is pretty powerful. So first of all, it seems that there's a lot of correlations to disease. It captures demographics and environment. So just as the example here is the Methylation Clock by Steve Horvath that basically shows that you can predict the age of a person just by looking at methylation, a methylation sample actually for every tissue, but it doesn't really matter. I take now a blood test or a saliva test, and I can basically tell you what's your age, plus minus 1 year. So it captures that the age very well. It captures sex very well. It captures, which maybe it's easy to ask about the age and about whether about the sex, but still it shows that there's basically a lot of information over there that is potentially useful. It also captures race and ethnicity. So it's essentially you can basically look at the first species of the methylation, and you'll see that there's relation to the ancestry. Then the third bullet here is that it captures genetics. So it captures other types of genomic information, including genetics. So there's a lot of variability in the methylation itself. So there's connections, correlations between the genetics and the methylation. And then there's correlations between the RNA expression and the methylation as well. And then maybe the most important thing in order to actually make progress here is that there's inexpensive high throughput technology. It's essentially the same technology as we do for genotyping, with just slightly, in principle, slightly more expensive, maybe $10 more. In practice, it's more, but just because the volumes today are much smaller. But if you think about 5 years from now, 10 years from now, there's no reason why it's not going to be exactly the same price as genotyping. The other advantage, which is more specific to the application that we have here, is that all you need is the DNA. And so if you have a biobank and you already have the DNA, you don't need to collect a different type of molecule. So RNA, for example, you basically just take the DNA and you measure the methylation over there. Okay. So this is the type of data that we get from methylation. We get essentially for every individual and every methylation site. So in a typical array today, we have about 800,000 sites that we're measuring. And there's going to be a number between 0 and 1. And that number would tell us what is the frequency of methylation in the sample for that individual in that position. So if we have 0.87 on the top left here, that basically means that we're in the sample of individual number 1 in that position. Let's say that's position number 1 here. There's 80, essentially you can interpret this as 87% of the cells have methylation. Okay, so that would be the interpretation of this. Of course, there's noise in the measurements and so on, but basically that's the if you think about abstractly what you're seeing here, this is essentially what you're seeing. So all the numbers in this matrix are going to be numbers between 0 and 1, as opposed to, let's say, genetics, where you basically have numbers that are 0, 1, or 2 that just count the alleles. Okay. So what we asked ourselves is whether we can use the methylation data just similar to genetic data. Can we use the methylation data for imputation of the EHR? Okay, so a very similar question to what people have been asking about PRS, about genetics, but now we're looking at methylation. Now, the problem is there's not so much data with methylation. If you think about genetics, there's hundreds of thousands of people that are being genotyped, or millions if you basically take all the biobanks together. But with methylation, that's not the case. And what's more is that it's not the methylation data sets that are out there publicly available. They're typically connected to a specific condition. So the phenotype data that you have is very limited. And what we wanted to do is we wanted to try to connect the methylation using the Atlas biobank to all the EHR data that we can get, which is essentially the EHR data from the UCLA. Okay. So why did we think that this could be something that would be potentially interesting? So if I look at the three points that I mentioned before about the limitations of PRS, methylation for each of those has something that could potentially do better. So genetics doesn't capture the environment. Methylation is affected by the environment. So we know this from different examples, including, for example, smoking basically would affect your methylation levels. Genetics is static, methylation is dynamic. We know that it changes over time, and that's one of the reasons why you can predict the age of a person. Okay, so this is something that basically, if we want to try to understand what's the situation, what's the condition of the patient today, methylation would give us information, kind of a pattern. The pattern of information will hopefully give us information about the condition of the patient today, including the information about genetics, but also the information about the environment and habits of that patient. Now, the third point, I can only say that basically methylation hasn't really been studied as thoroughly as PRS, and that's kind of an understatement. It really, I mean, just the numbers are basically a few order of magnitudes smaller than what you have for genetics. So the potential, that doesn't mean that methylation is going to do to get us much more information than genetics, but at least it gives the potential here. Basically, we have, if we do find something here, it could be potentially much stronger signal than what we have in genetics. So we wanted to check this and to look at this hypothesis and see if we can get more information here. Okay, so we tried something that we call Methylation Risk score. We thought that we came up with the name, but actually it was used before in a few papers. But basically, the idea here is exactly like the polygenic risk score. You want a linear model. Now you have these effect size inside this beta I. In genetics, it was sum of beta I GI. GI is the genotype. And in methylation, it's sum of beta I MI, where MI is the methylation at position I in the genome. And we measured whole genome methylation again on the Atlas population. So this was over 831 Atlas patients. In fact, we looked at more, but basically after exclusion, we got to 831 patients. And then we constructed MRS, so this methylation risk score for 607 phenotypes, including so ICDs, and so it's essentially the disease conditions, diseases, medications, and clinical labs. And again, there was some exclusion and inclusion criteria here based on basically how rare is the condition. We can't really predict something which is very, very rare. And then to find these effect sizes, beta I, we used regular regression. So this is basically regression with penalty that could be either L1 or L2 penalty, so either elastic net or lasso or ridge. And then we compared it also to a baseline model that basically just used regression over age, sex, BMI, cell type proportions that we could calculate from the methylation, ancestry, smoking status, and then the 10 top PCs of the genotype data. So that should give you the ancestry. So that would be basically the baseline. And the baseline here, as you can see, it also uses some of the information from the methylation. Yeah.
Technical question about the cell type portion. You say you use the methylation. Can you also just, how does that compare to the, you know,
yeah, CBC, so we didn't have CBC for all of them, but basically it's pretty high. There's the Hausmann method, which is the one that we use here, give you very high correlation to that. And so yeah, but in one of the things that's actually bringing me to that the comment that I wanted to say here, which is we use here the 10 top PCs and the cell type proportions that you sometimes don't even have, right? And so we actually give the baseline a little bit more kind of just a little bit advantage over what you'd have in kind of a regular baseline. Okay, so that's the demographics of the population that we have. So maybe one thing that I would mention here that it wasn't mentioned in these two slides is that we had kind of the idea of how we have a little bit here of, not a little bit, maybe a lot of ascertainment bias here on how we chose the population. So I should just kind of, for transparency, basically, I want to kind of explain a little bit on how we chose these patients. What we did is we used the random forest algorithm to basically try to predict from the EHR data whether a patient is going to have a risk for acute kidney injury after surgery. So all these patients went to surgery, and we wanted to basically choose patients that had risk for acute kidney injury post surgery. And we took patients that had high risk and then had AKI, and then patients that had high risk and then didn't have AKI. So that was kind of the design of the study, which essentially means that we're going to have some enrichment for some specific conditions that, for example, are related to kidney conditions. So, which essentially boils down to basically having more power to do better prediction for those. Okay, so, but other than that, you can kind of see here that there's nothing very, out of the ordinary in that distribution. The distribution is relatively, so the patient population is relatively on the kind of skewed to the right, which makes sense if you think about basically both risk for AKI and they go to surgery. But males, females, there's a bit more males, but basically it's roughly 50%. And we do have some diversity in terms of the population. We try to increase that, but basically we didn't have enough samples at that time in Atlas to basically go to larger numbers of other populations, so the Latinos and Asian and Black. So the majority are Caucasians, but we do have some diversity here. Okay, so this what you see here are the results for fee codes, which is essentially basically you can think about it as ICD codes, so diagnosis codes. They just grouped medications and labs, and in each of the figures, let's look at this figure here, for example, on the left side. So the x-axis would give you the performance on for the baseline algorithm, and the y-axis would give you the performance for either the MRS or the PRS. So you compare the methylation to the genotype data, and so you'll have a point for the MRS and a point for the PRS for each of the conditions. If you're doing essentially this roughly the same as the baseline, you expect everything to be across the diagonal. If you're doing better than the baseline, it should be above the diagonal, right? And so you see that for the MRS, we're pretty much always, arguably always above the diagonal, maybe sometimes around the diagonal. For the PRS, sometimes we're better, but most of the time we're actually around where the baseline is. I'm going to give a little bit more context on this in the next few slides, but basically this is something that you also see for the medications, and you also see this for the labs. So maybe for the labs you see actually some better results for the genotype data.
Are you using an out of this sample, like for medication, is there PRS related to medication?
Yeah, that's a great question. That's basically bringing me to the next slide that I'm going to talk about. But I'll give you the short answer here, and then we'll get back to this. This is actually PRS basically within sample. So one of the issues here could be just the sample size. So you don't really expect the genetics to be a great predictor. But the point that I'm trying to make here is that for the methylation, you don't need 500,000 to be able to get really great results. I mean, I don't know if these are great, but better than the baseline. For the genetics, you definitely do need. So 1,000 or 800 is not enough. So what you see here in the bottom ones, you see an example from the fee codes, an example from the indication, an example from the labs, where you could basically, so just 3 examples. And for the phi codes and the medication, it's essentially it's a binary a prediction. So you're trying to predict a binary outcome. And what you can do is look at the deciles of the risk PRS or the MRS and see what is the proportion of the cases inside each of these deciles. And you hope that you're going to have a higher proportion of those when you go to the kind of higher risk PRS or higher risk MRS. And you see that we get, at least for this example, we get really strong results for the MRS. So you get about 80% of the people that are, of the patients that are at the top 10% of the MRS score are actually cases. So this is, so if you went only, if you decided to go only to the high risk MRS patients, you'll have a pretty good precision here. And the same thing happens with it. So this is actually true for these two. And then for the right one, you basically, this is a continuous, you're trying to predict a continuous outcome. And so essentially it's basically you're looking at the observed compared to the predicted. And you hope again that it's going to be y equals x kind of thing. So this is going to be correlation 1, and you see that the methylation, not perfect, but it gives you a R square of 0.5. And with the genetics, we almost don't get anything here. So yeah, so one of the things that actually came, this is still basically within samples, so with these 800 individuals, but one of the things that we actually got from the reviewers is that, you know, maybe the problem is that we don't have imputed data. So we didn't impute the genotypes. And what we show here is that actually that doesn't really matter. You impute the genotypes and then you do a penalized regression, you basically would get essentially the same, about the same results. Now, to your question then, this is exactly the slide that basically kind of answers this. We asked ourselves, okay, so what happens if we take PRS that is computed in the UK Biobank, for example, and then we apply it here? It did do better if you can look at these 4 different things that you see here. So the UK Biobank PRS is the first one, the MRS is the second one, the PRS is the third one, and then the baseline. And you can see that in many cases, the UK Biobank PRS gives you better result than just doing the genotypes. So the PRS based on this small sample. And the confidence intervals are relatively large just because our sample is small. So we can't change that at this point. But the methylation is definitely much stronger than all of them. Okay, in this example. Now we couldn't do this across everything because it's not that everything that we have in the EHR data we have a PRS from the UK Biobank, right? But basically what we did is we looked at a set of those where we had kind of the overlap between those two. And one more comment here, that this PRS that we use here for the UK Biobank is actually also using penalized regression. That's coming from a group in Stanford, actually multiple groups in Stanford that basically worked on this, including Manu Rivas and Pritchard, and I forget who else is there, but Tipshirani and Hasty. So there's also the statisticians in the mix over there. So it's, you know, we believe it's pretty solid. It's not that we took kind of like the worst PRS in the literature. Okay, so then the next question that we can ask is, so this is methylation. This basically gives us the information just from the methylation. The MRS or the PRS is just the methylation and just the risk from genetics. However, if you basically look at what people have been doing before, you know, or what we are doing essentially, if we only look at the EHR data, we don't have any methylation, we don't have any genetics. What do you do then? So there's a bunch of methods that people developed to do imputation from the EHR itself. So you only take the EHR data and you try to impute the EHR And one of the methods that is kind of very widely used is using matrix completion. It's called soft impute. And we asked the question is, can we use the methylation to complement the matrix completion method? So we're not kind of trying to compete it just methylation just in compared to just the matrix completion, but we're looking at the matrix completion plus the methylation compared to just the matrix completion. So does it add value beyond just doing the matrix completion? And there's many different ways that we could potentially do this. We just did the kind of version 0.1, which is kind of a very simple approach. The approach is basically you first compute the methylation risk scores and you get, so if this is basically, you have some missing data here, now you have some information that is imputed by just the methylation, but now the matrix is actually complete, but maybe not very accurate in some cases. And so now we concatenated the two matrices, one next to the other. So the matrix that basically has the missing data and the matrix that is computed from the MRS. And we just feed it into the matrix completion to soft impute. So we are actually using now the matrix completion. So we didn't do anything fancy in terms of the algorithm. We basically just did a two-stage approach here. And what we get, so now we get a prediction or imputation, and what you see, for example, in this position here, we had from the methylation. So this is obviously a toy example. I kind of invented the numbers here, but let's say we have a 0.2. So the methylation would say that there's a 0.2 probability that that person has type 2 diabetes. And then the next stage, basically, we can use this information to get to just impute 0 in this case by the matrix completion. So we looked at this, and what we can see is that in many cases, actually in almost all cases that we look, there's an improvement when you look at the soft impute at the matrix completion plus the MRS. Sometimes it's not a huge improvement, but just keep in mind that these are just 800 samples, so we are limited by the size. But across the board, basically, there was an improvement. And sometimes the improvement was statistically significant. Or actually, in many cases, it was statistically significant, but in some cases, the effect was actually pretty big. Okay, so the other thing that we wanted to check is that, you know, when you test the methylation and you look at the let's say clinical lab, the question is typically they're not going to be done at exactly the same time. And the question is, how much do you lose by doing the methylation 3 months after you did the clinical lab? And from everything that we looked at, there was no effect when we looked all the way back a year back. This is actually the only example where we saw that there is a statistically significant difference across time. But, you know, as you can see from the picture, the effect is not huge. And again, you have to remember that our sample size is not very big. So there could be other cases where it's statistically significant, but probably the effect is not very large because otherwise we would have seen it already. Okay. Again, taking this with a grain of salt because the sample size is not huge. When we looked at the difference between the MRS across the different populations, we didn't see any statistically significant, not any. We saw actually we checked 180 comparisons, and 7 out of the 180 had a statistically significant difference. But for the most part, there was no big differences between them. That again could be explained by the fact that the sample sizes are small, so it's not statistically significant, even though there is a difference between them. But yet again, if the effect is very large, you'd find it also with a small sample size. Okay. One thing that I think is very important to, and I kind of try to emphasize it in the last few slides, is that we have a small sample size, and we can basically ask ourselves, what would happen if you increase the sample size? So you can sample and see how do the results look as you sample. And what we observe is that if you look at basically the baseline algorithm, so again, this is just the regression over a very relatively small, let's say 10 features. 15 features maybe. This is roughly constant over the sample size, at least the range of sample sizes that we looked here. But in our case, in some cases, there's a very clear increase in performance as you increase the sample size. Now, we only looked at 800. It's not clear to me that we need 500,000, but definitely if we'll go to 5,000, 10,000, we'll get much better results just by looking at these figures. Okay, so this is essentially, I have another 10 minutes. Oh, yep, okay. So I want to give just a quick overview of a second part that we basically, it's kind of actually in process. We're basically building it right now. So it's going to be very quick, but just to give you kind of a glimpse into this. So a couple of years ago, we had a paper that basically tries to do a deconvolution of methylation data. And the basic idea is that if you have multiple cell types, and each of the cell types has a different level of methylation, what you actually see when you look at the bulk data is kind of a combination of multiple cell types at the same time. So I'm going to skip some of these slides just to go directly to what I want to say here. In this case, for example, if you look at the cases in the controls on the third cell type, you'll see that there's a difference. So the kind of the brightness is basically how much methylation there is in that cell type. Okay, so you see that they're much darker in the cases compared to the controls. But when you look at the observed data, which is the bulk data, that's for every person, there's going to be a different mixture of the cell types based on the cell type proportions of that individual. And then you lose a lot of this signal, and you're just not going to see, unless you have a much larger sample size, you're not going to see the effects that are basically kind of hidden in specific cell types. So what we did is we tried to think of ways of going from the observed bulk data into each of the cell types. And what we were able to show is that if we kind of came up with a method that I'm going to skip today, but basically we came up with a method that if it was perfect, then you would not need any cell sorting or single cell sequencing. Obviously, it's not perfect, so you still need these other things, but basically when you don't have them, you can still use this deconvolution. Again, I don't think I'll have time to go into the details, but I just want to make sure that it's clear. It's very different than what sometimes people call deconvolution, but there are really decomposition methods where you take the matrix of the individuals by the methylation levels, and you try to decompose it into two matrices. One of them is for each individual, you want to know what are the cell type proportions, and the other one is for every cell type, what is the typical methylation that you have in every position. So this is what you see at the top here. And this is if you know the Hausmann method, for example, that's what it would do, but also PCA, essentially, that's what it does in an unsupervised way. What we're trying to do with our method is called TCA, tensor composition analysis, is for every individual, we'll have a matrix that will say what is this for every cell type and every methylation site, what is the value that we have over there. Okay. And then on top of this, we'll have for every individual, we'll have the cell type proportions. Now, this is, if you think about it, this is a bit of a magic because the input data that we have is individuals by methylation. And so the number of individuals by 800,000, let's say, and the output is for every individual, we have cell types by methylation. So we have number of individuals times number of methylations times the number of cell types, which is basically larger than the input. So that actually doesn't make sense, right? So if you individuals times methylation sites, that's the input. If you have 6 cell types, you'll have 6 times the number of individuals times the number of methylation. So essentially what you're doing, or what we try to do here is to invent data, which is impossible. But it is, the argument is that it is possible if you're making some assumptions on the distribution of the data, the underlying distribution of the data. And unfortunately, I'm going to have to skip this, but this slide tries to give kind of a little bit of intuition of why this is potentially possible under some basic assumption. And then the question that you can ask is whether these assumptions are reasonable or not. The way I think about it is that the proof is in the pudding. If you can basically do prediction using this, that means that you did that at least some of these assumptions make sense. Again, maybe not perfect, but it gives you some information. So I'm going to skip these couple of slides that explain the model. I'm just going to mention that in that original paper, what we did is we used it to find new associations that are cell type specific association, and we replicated those on sorted cell types. So again, kind of like that's kind of a proof that basically the assumptions that we made make sense, even if they're not perfect, they made sense. And then what we did here is we tried to use, so think about it this way, you take an individual, right now we have 800,000 positions, and now we basically multiply this by 6, because now we have it for every cell type, and we run regression on this 800,000 times 6. And we can call it CMRS, cell type specific methylation risk score. And what we see is that basically, just generally, maybe I would say almost in all cases, there is some improvement. Sometimes it's more substantial than in other cases. So what you see here is the difference between the MRS and the CMRS. And so these are just a few examples where you see that there's a CMRS gives much better result than just the MRS. Okay, so I'm gonna skip those, and I'm gonna go into the conclusion. So basically, what the main conclusion that I hope you're convinced now after seeing this talk is that methylation has a lot of power that is basically unrevealed at this point. So we have or understudied in terms of potential for applications in clinical applications in terms of imputation, or ideally, we haven't shown it here, but ideally a prediction of what's going to happen to the patient in the next 5 years, right? So that would be one of the next steps, because what we showed now is basically imputation, what happens right now to the patient. But the question, the much more interesting question, I think, is what will happen to the patient in the next 5 years based on the methylation today, right? So we haven't shown this because at the time that we looked at the methylation, we have methylation for that. We didn't have the 5 years, but I guess now we do. So we could basically do this as one of the next steps. And so again, the main thing here is that it's really understudied, and the sample sizes are much smaller. And I would advocate for basically adding more methylation to the biobanks, hopefully for Atlas, but also to others. And because as you can see here in the figure on the bottom right that I showed before, sample sizes are really not getting to the, we're not using the potential of methylation yet. And I would just mention a couple of applications here that even for what we did so far. So one of them is EWAS confounding. So if you think about epigenome-wide association studies, one of the problems that you can have in these studies, so in these studies you compare cases and controls with methylation. One of the problems that you can have is that if there is some confounder that basically appears in the cases, doesn't appear in the controls. Let's say all the cases are taking a medication, if you don't have the measurement of that medication for these patients, you don't know that that's what they're taking, you're going to find a lot of false positives. And using this method, you could actually add the predictor of whether the person basically took the medication or didn't take the medication. Same thing for other confounders, smoking and other environmental factors that go with. So this is something that we're working on trying to show that in some cases you can correct the EWAS using these kind of approaches. A second one is medication adherence. So basically you want to know if a patient is actually taking a medication, because 30% of the patients that are prescribed a medication, they don't necessarily take it. And then some future directions. So one of the things that I discussed here, just the correlation, just the prediction, but there is no causation here, or at least there is no, we haven't discussed causation. And it would be interesting to basically try to understand what drives these predictors. And so what's actually under the hood over there. And then the second thing I mentioned, this improvement to soft impute to the matrix completion. But what we did was really kind of a first step, really at version 0.1. And we just wanted to evaluate if there's a signal over there. And the question here is whether we could do something better. And then, as I mentioned, population size is limited. So let me just finish here and say that so the TCA paper that I mentioned that was led by my student Elior Hoffmani, who's now in Berkeley, postdoc, and the colored individuals are basically people from the lab. And then the MRS and the CMRS works are basically by a few students from the lab, including Mike Thompson, who's here, and Noah Zaitlan from here from UCLA has worked with us on this. And Brian Hill and Johnson Chen were also involved, Johnson specifically in the CMRS predictor. So this is actually it's not published yet, the paper, or some version of it is basically in the archive, but it's hopefully soon is going to be in the formal press. And then again, thanks for all the funding and specifically to, so this work was done based on the Atlas and that, was available through the Institute of Precision Health and working with the Atlas team, the IPH team. And it was funded by a seed grant from CTSI. So it's great to give back by giving this talk. Thank you, everyone.
Super interesting. I think we have some time for some questions, and I'll take from the audience because people here came. So, yeah, Jason.
So have you tried taking your MRS and finding relevant EWOX studies and seeing if you can predict sort of out of sample traits in these EWOX studies or vice versa?
Yeah, so we did just a few replications like this. And that's basically something that the reviewers actually asked us to do, and we and that seems to replicate when you go to the outside world.
Without any like losses?
No, no.
It's interesting, you were talking about sample size.
Mm-hmm.
And it seems that with your sampling strategy of seeing how much power you get or AUC improvement with each addition of a quartile or whatever, you should be able to kind of follow that out. And for many of these, make an assessment of what's the sample size that would give you, where does it start to asymptote? And I'm assuming that during your talk, you were mentioning, oh, we need 5,000, 10,000, that it's somewhere in there, but I'm wondering if you guys have done that formally.
So, yeah, I'll just repeat because I don't know if people heard on the Zoom, but basically the question is if we can estimate what is the sample size that we need to basically get to kind of where it plateaus, essentially.
Yeah, which you should be able to do from your down sample.
So I think in some cases we could, because think about the graph that actually I'm showing here. So in this graph, we're actually just in the beginning of the slope, right? At some what you expect is something that would go up like this, and then at some time we'll have kind of marginal improvements, right? So it's going to plateau at some time. We're pretty far from this, at least based on for this specific one. For other ones, we may be closer, and so we know that probably if we had another 5,000 or 3,000 or whatever, we can have a specific number for each of these things that we're predicting. But for things like this, so for chloride here, we don't know, right?
Well, how about if you just assumed it's going to go up monotonically because it looks monotonic there?
Yeah, I mean, so you can assume, but it can't go monotonic forever because it's bounded by one, right? So
no, at some point it's going to hit one, and so
yeah, you can bound it by that. That's true. So basically, I mean, we can do the calculation right now. So you go from 5%, let's say, to 15, so 10% for every 100%, every 800 samples that you take, right?
So 8,000.
Yeah. So it's
I mean, that's what I'm trying to get at, because it seems that, like you were making comments about having 5 or 10,000, it seems that having 5 or 10,000 is going to give you a lot of power for a lot of things and be pretty close to diminishing returns.
That would be my intuition, right? But we haven't we haven't really formally done this, so I can't. But basically, just looking at these figures, that's what I would guess.
So it looks like, let's see, there's another question there. Although other alternatives for methylation is additional consent needed No additional consent is needed. I'll
So-
answer that.
So yeah, no additional consent is needed. And then the first question was, are there other alternative than methylation? So yeah, there's actually methylation is not specifically special in the sense that in terms of dynamics and all these kind of things that basically we discussed in the beginning, but it's relatively easy to work with because again, we only need the DNA that we have it already in the biobank. If we wanted to do it on RNA, we had to collect additional samples. This way, we basically just decided, okay, we're going to look at these 1,000 samples because of these criteria, and the samples were there, the DNA samples were there. You could potentially look at microbiome, you can look at other things that could potentially give you the same type of idea, but then you need a biobank that has the microbiome or a biobank that has the RNA. So other than that, I don't really know of something specific that would say methylation is better than the other ones. You could maybe argue that methylation changes over months and not over days or not over, so there's kind of the time that it changes is an advantage if you want to capture the information about the patient in the last few months. But it's not that these are kind of more intuition and gut feeling.
Well, thanks. I think we've hit the end of our time. Looks like there's actually another
Okay,
group coming in, so we'll.
thank you, everyone.
Yeah, thank you. Great.